morning, everyone, and welcome to today's webinar. It's hosted by Mass Robotics Healthcare Robotics Working Group. My name is Juan Necochea, and I'm Director of Strategic Partnerships at Mass Robotics. We're super happy to have you here, and we will dive into the fascinating world of healthcare robotics with our esteemed collaborators in just 1 second. At Mass Robotics, we pride ourselves of being the largest independent robotics hub dedicated to accelerating the innovation and adoption in robotics, automation, and embodied AI. Our mission is to foster the development of successful technology companies by providing them with essential resources, workspace, and valuable connections. Our working groups are open to the entire robotics and automation community. They're designed to meet industry needs and drive the adoption of robotics for both commercial and societal benefit. I encourage anyone interested to reach out and get involved. We currently have working groups in various sectors, including drones, AMRs, autonomous vehicles, and of course here, healthcare, in addition to others. Please check them out. I'll follow up after this event with some details on how to participate on that for all attendants, I mean. And I also want to highlight the Healthcare Robotics Startup Catalyst Program, which is developed in collaboration with Festo, Mitsubishi Electric, Novanta, and MITRE. I am being internally, I am being corrected here that indeed we are talking about, when we talk about embodied AI, we're talking about physical AI as well. The Catalyst program supports healthcare robotic startups by offering them mentorship, access to resources, and opportunities to connect with key industry stakeholders. We have selected an incredible 2025 cohort. This is our 4th cohort, and it's comprised of a super diverse group of startups, many of whom are logged into this webinar here today. We'll be holding an intro session to introduce you to that cohort on February 6th. So make sure you reach out to us if you're interested in participating in that meeting. Our working group already will be invited. So let's dive into today's activity. So today we'll hear from our featured speakers. So we have here Johannes Linkbach, who leads the Research Hub Boston in Life Sciences for Festo. We have Ross Gallant, who specializes in the industrial segment for the Life Sciences group at Festo, and Sid Gupta, Research Electrical Engineer at Festo USA. I will moderate the Q&A sessions, and then after the presentation, we'll kick off that session, and we'll do our best to ensure that everyone has a chance to be heard and engaged. There is a section in the GoToWebinar where you can provide questions in the chat section. So as we develop the webinar, feel free to drop in questions. They don't have to come in at the end. We welcome you to kind of, as you are kind of bubbling the questions, drop them in there, and then we'll compile them towards the end in a Q&A session so that your questions are addressed. Before we begin, a few housekeeping notes. This webinar is being recorded and it will be available publicly after the event on our Mass Robotics platform. As mentioned as well, if you have any questions, please feel free to type them in the chat and we'll address them during the Q&A session as time allows. Now, without further ado, let's dive into today's session. I'd like to hand it over to our first speaker, Johannes. I hope you enjoy and I encourage you to engage in this dialogue. Thank you.
Amazing. Thank you, Juan. Yeah, welcome everybody. Really nice to have you on the call today. We were looking forward to it, to give you an insight today in the webinar on challenges we see in the life science environment, but also of course have an insight to the solutions we are providing. And yeah, I hope this is insightful. That was the main thing we tried to cover here. And so in the first part of the presentation, Ross will give an overview of our current portfolio and what kind of solutions we see in the lab automation and life science overall environment. And also interesting here might be in particular the approach we are taking in terms of our cell structure. That's the takeaway from this part. The second part, Sid will guide us a bit through the topic of liquid handling with different dispensing and pipetting solutions, making the differences, showing like what challenges are in there and which technologies help us to cope with these challenges. And in the last part, I will share a bit of our future technologies where we see that technology might actually help in one of or some of the main challenges which we see in the life science environment. And yeah, that's like where we want to take you today. And we will always have some, Ross, please click to the next slide in this presentation. Thank you. And let me start. We will keep it brief, but for everybody who doesn't know Festo, we will do a short introduction. So Festo is a 4 billion company, globally active, and we are one of the leaders in automation. Yeah? And automation here is pretty broad. Yeah? So everything what's about like with motion technology, whether it be like physical goods, for example, but also liquids or driving chemical processes, this is like what we try to cover or what we are covering today. We're like active, as I said, all over the world, like over 170 countries we are in there. And with our over 20,000 employees, we have a broad network of distributors, of course, but we also have a quite significant portion of R&D, which we're covering. So Festo is a family-owned company with a pretty nice outcome in patents each year. And this is due to also the reinvest in the company. So like about 7 to 8% of the yearly revenues are being reinvested into R&D, which helps us to also get into new automation challenges in almost any industry we're working in. Ross, if you go one slide further, please. Yeah, and to give you an impression, so our portfolio has mainly two main values we bring here. On the one hand, our standard business, everyday business, is automation. And we have a broad portfolio of pneumatic components which are deployed into various industries, whether it be automotive, food and packaging, or semicon and electronic light assembly. Then we have a broad range of electrical automation. This means like if the application requires it to get or to switch technologies here, this is also something we provide. And then we have a larger range of products for liquid and gas handling, which is basically also the most important one for deploying into the life science environment. So we are an expert in liquid handling, like moving liquids from A to B, providing filling solutions, for example. And then in the end, we have customer solutions which can range from assembly to order up to engineering to order, which means like we are actually building products together with you, or like subsystems which can be integrated into machines. And the important part here, Festo is a component manufacturer, so we are not building the entire machine, but way more, we are enabling automation companies to drive their processes as efficient as possible. And in addition, we have a didactics business, which is mainly focusing on vocational trainings, which is like for the industrial environment to get people ready for solving exactly their automation challenges with, of course, Festo products. All right. And with that, I would love to open up the stage for our today's topic, way more specific for the solutions for life sciences. And the purpose which drives us, the mission here, is definitely advancing human health through automation. And yeah, really looking forward to the upcoming presentation. And with that, Ross, it's your part.
Hello, everyone. My name is Ross. I'm coming to you live from our Technical Engineering Center in Marlborough, Massachusetts. I'll be discussing the applications of laboratory automation and diagnostics. So Festo has about 32,000 components and is developing new products every day with inspiration from industry. Today we'll focus on the core life science applications. Some of you may not be familiar with the technology, so I'll go into a brief example for clarity. With sample handling, it consists of 3D gantries, end-of-arm tooling, automation software, and sensors for precision and reliability. In vitro diagnostics are tests conducted on samples taken from the human body, such as blood, tissue, and other bodily fluids to detect diseases and conditions, as well as infections. In vitro, like in vitro, point of care diagnostics are tests done at a faster process, normally close to the patient, and cuts down on lead times for samples to reach the lab and deliver results. These are normally tabletop solutions that do not scale up. And then finally, bioreactors are manufactured devices and systems that support biological active environments. This provides a suitable regulated environment for cell growth needed in research and development. Each one of these applications has its own set of challenges, which we will discuss. So Festo tackles these challenges by having a hybrid component supplier model, as Johannes described, using our customer solutions team. Companies now need Festo to be there throughout the customer lifecycle for consulting phases, operational phases, and maintenance phases. Festo is a very powerful engineering capability, a worldwide production network, and a logistics infrastructure to support development on a global scale. With this model that you see here, we're equipped to design new undeveloped technologies along the experts in industry. So some of the examples of how we develop technology will be shown in the next few slides. In this example, we were asked during the beginning stages of COVID-19 shutdown to integrate an end-of-arm tool that could aspirate and dispense on a pre-designed system. Additionally, we would need the end-of-arm tool. We would need an additional end-of-arm tool that was compact and capable of gripping and decapping sample vials, which hadn't been created yet at the time. The challenges here were precision, reliability, and increased throughput of tests per hour. The previous process was done by hand. It was very slow and risked human error as well as cross-contamination. So with software automation and custom designed components, we were able to achieve a system for the customer that increased throughput and accuracy from tens of samples per hour to over 320 samples per hour. So here, the real challenge and success was creating a very compact rotary gripper that could decap, cap, and grip as well. In this next example, we helped reverse engineer an in vitro diagnostic process that analyzed DNA and designed a smaller mobile point of care system that was made up of about 90% of Festo components. The original process was stationary and had a much larger footprint, which we were able to reduce to about 2 feet by 4 feet, as well as making the application mobile. The project highlights the importance of product synergy, as well as the additional design services offered by Festo, which is a standalone component supplier. As any other standalone component supplier, this is very important, and not many companies can achieve this other than Festo with our hybrid model. In some cases, customers want a stationary industrial solution, but labor costs and operation can be expensive. In this example, Festo was challenged in creating a continuous process from start to finish that was completely automated. Many of the components here were used, including dispensers, decappers, grippers, AI-driven optical scanners, and conveyors, which were all integrated with automation software. The solution reduced the cost, labor size, and streamlined the efficiency of extracting DNA samples from different vegetation to enhance the development of resistant seeds in agriculture. And another challenge, and something that was inspired by industry, was a process of cellular growth for vaccines, gene therapies, and lab-grown meat via bioreactors. Although the process has been perfected in large-scale production, the most cost-efficient way to improve this process would be to scale it down and provide a smaller batch and smaller footprint for the R&D labs who are limited by space and costly research real estate. Knowing there would be an increased need for this in the future, Festo invested in designing new components such as piezoelectric-driven valves, mass flow controllers, and vacuum pressure generators to aid the process of creating tabletop reactors, bioreactor systems that achieve optimal cellular growth. With that, I'll now hand it over to Sid to give you an in-depth look at the components that were developed through the industry needs.
Thank you, Ross, for giving a vivid picture of the applications. Hi, everyone. My name is Siddhant. I'm the electrical engineer for research. at our Marlborough facility. So as Ross already explained to you, the 4 areas for life sciences, biopharma, or laboratory settings in general, where we have deployed some of our solutions. So when it comes to sample handling, IVD, point of care diagnostics, bioreactors, we also need to break it down to some fundamental building blocks and talk about other aspects of automation where we may face challenges while commissioning such systems. One such aspect is handling the liquid itself. It could be the high throughput of buffer solutions required for sample handling. It could be non-invasive analysis of samples in IVD and point of care diagnostics. So first I'll get into what's liquid handling in general. So liquid handling means the precise manipulation and handling of liquid volumes in a laboratory or a clinical setting. Uh, this could go down to small fluid droplets at the microliter or nanoliter level uh, for preparation, experiments, analysis, recording data, and so on. Liquid handling is classified into dispensing and pipetting, and I'll be giving a traditional picture into dispensing and pipetting and how we need to automate some aspects of these processes to get rid of the challenges that we have at the industrial level. Next slide, please, Ross. So dispensing. Dispensing is a pressure over liquid process. It can be a contact-based or a non-contact based process. So what you see in the schematic on the left is a 2x2 valve, which means it has like a fluidic input and a fluidic output, and it can be operated as normally open or a normally closed valve. So it is generally used for dosing buffer solutions in large quantities while preparing, let's say, for example, cell cultures or initial batch of materials for starting an experiment. And on the right, what you see here is the use of some of the components from our Festo portfolio. For example, our pressure vacuum generator box, the solenoid Vica valve, which can be controlled using a valve driver. We have a flow sensor and a pressure sensor of the flow through type to keep a check on the process. Next slide, please. So similarly, pipetting is a contact-based technique where we aspirate a specific volume of solution at one source container and deposit this solution to another target destination by using a permanent pipette tip or a stationary metal or plastic pipette tip. It could be something like hypodermic needles. We are also using disposable tips, which can be used to reduce carryover or cross-contamination between different samples during the dosing process, especially useful for something like handling patient applications. For example, where we have a PCR sample from a patient, and of course, it's unique to each patient that we are working with, and we want to minimize carryover as much as possible. So the use of disposable tips in such a scenario is the most optimal way of dealing with the samples when we talk about traditional pipetting as such. Next slide, please, Ross. So, however, we have seen during various situations, especially what we also saw during the pandemic, that we need high throughput through these systems. We need increased precision and accuracy, which is one of the quality check parameters for these processes. Minimized contamination, we already touched upon during dispensing and pipetting, as well as the flexibility to integrate various dispensing components into the overall modules that we use for dosing. So here you see a multi-dispense head Festo module in action, where there's the filling of a well plate. The lowest that we see here is 4 microliter per shot. Next slide, please. So first, talking about dispensing systems, how do we solve the needs for automation in dispensing systems? So we have these uh, multi-channel dispense heads at Festo, where we can go from a few microliters to milliliters, and the internal components, including manifolds, valves, they are media-resistant materials as far as both aqueous and non-aqueous solutions are concerned. The individual configurability of each of the dispense head is something which sets these modules apart, because each dispense head can be configured for a different pressure, different actuation time, volume, and so on. Easy integration is extremely important, especially when we are working with a multi-domain, multi-specialization industry. Our users could be biologists, somebody who's an expert with PLC-based automation, or a fresh student out of university who's very used to the Python environment. So we have drivers for each of these devices to cater to the users, and they can be integrated easily into their overall automation workflow as far as their code base is concerned. One example where we are modularizing the overall dispensing process is through providing analytical quality check using AI, uh, so that we get tighter dosed volumes as well as precision between multiple shots. So this problem in overall, if we look at it, so in a dispensing system, we have the modular dispense head may be mounted on one of the movement axes. There's the environment around it. There could be temperature, humidity, other changes. So there are multiple variables in this system, and it's difficult to have a completely bound mathematical problem around it, uh, where we have some control variables. So there's always a part of the system that way which cannot be modeled using state space equations. So here comes in using a machine learning model, something as simple as a fully connected neural network, a simple 4 or 5 layer neural network, which takes in the known system parameters, such as the size of the reservoir, length of tubing, size of needles, so on, and sensor-based proprioception. So sensor-based proprioception means that we use sensors to read the internal state of the system to give us some primary variables as well as some derived variables. So primary variables could be just the temperature of the fluid flowing through the lines. A derived variable could be viscosity based on this temperature. Other variables like turbidity of the fluid flowing through the fluidic lines, and so on. So once we have these training data, we go for something like a feature extraction algorithm on this data to extract the most important features, which we see could be a trend for the machine learning model to train on. This also reduces our memory and computational utilization overhead. So we don't need to utilize entire time series data from any type of sensor that we use in these processes. So finally, after training the model with respect to a ground truth, which validates for us the volume being dosed, we are able to predict the volume which is dosed for each shot, as well as improve the accuracy and precision of the dispensed volume as we go from one shot to the next shot, provided we have sensors with higher range of the response time in the millisecond uh, range. We have media resistant sensors like the other media resistant components that we have in the manifold, as well as a fast responding DAC system, which can operate at a high sampling rate. Next slide, please, Ross. Yeah, similarly, so we can also have modular systems for pipetting. These could be single channel systems, multi-channel systems. What makes us differentiate over here, or what makes our life easier here, is that each of these pipette manifolds uh, have been designed in a manner that we have very low dead volume within the space itself, and also they can be configured separately as we operate them using a valve driver such as RVAEM or any other digital signal. We can use digital signals such as over the RS-485, RS-232 protocol, which in turn causes the valve drivers to send out something like a PWM signal to control each of these pipette heads. This minimizes cross-contamination and carryover between samples, as well as we are able to work with any disposable tips, be it from us or any other third-party standard sized tips. Next slide, please. So this is something that is right at the edge of innovation in liquid handling. We have this new group of users who are specifically working with microfluidics in the context of lab on a chip. So what you see in the video here is a microfluidic chip which has emulsion droplets flowing in it. So in the traditional context, if we look at a few years back, for setting up something like this, they would have to just take a huge bulky solution with an overkill in terms of the pressure being supplied, in terms of the number of valves in the manifold. It could also be things like those systems would only work in conjunction with a PLC system for control using industrial protocols like EtherCAT, Profinet, etc. So a lot of these requirements or features were an overkill for our biologist user in the academic market, or for even startups in the biopharma or the life sciences space. Uh, so what we developed is a modular approach to tackling such problems. We are able to use our standard Festo components and provide the user reconfigurable solutions as they build up expertise within the domain of lab on a chip. They could like dose using a single pipette head into the chip, or they could also work with a multi-channel chip along with a multi-channel pipette head in case they need to produce emulsion drops with multiple reagents. So this has been something that is I wouldn't boast, but I would say, at least from the perspective of an electrical engineer, this seems like very groundbreaking. And what we have seen here is that the flow rate control, the volume control, uh, irrespective of the viscosity of the solution moving through the chip, uh, is something that is at par with other systems being used by our target user group, as well as the flow rate is high enough to avoid sedimentation of particles within these drops. An example use case of such a chip could be non-invasive analysis while using cell-based therapies like CAR T cell therapy. Uh, for example, in a bioreactor, it would require some invasive sensor uh, to observe the process. However, here we can be non-invasive while while also being sterile during the analytical process. Thank you so much, everyone, and I would hand over to Johannes.
Amazing. Thank you, guys. So we come to the last part of the presentation, and we want just to give a bit of an insight what's driving us at the moment. we've been a bit selective on like the topics here, as well as for the technology to show later, but it might be a nice insight, or I'm sure it's a nice insight still to see what's ongoing. So what we're at the moment, what we're recognizing, of course, is like the increase or the beginning of the cell and gene therapies, a lot of tissue and organ manufacturing, which comes up, which places interesting questions to us as a component manufacturer, which goes from like on the one hand, for example, handling cells, being able to produce in a sterile manner. These are all things we're assessing at the moment, but also the question of minimizing sterile environments or volumes in a process, for example, is one topic, and I will present one technology which is offering a solution for that at this point. But we also see a lot like healthcare automation, in particular from the working group here in Mass Robotics. We had wonderful use cases this year, last year also, which were leading the way into like automating dangerous and exhaustive processes, which are actually dangerous for humans to execute. And then, of course, more in the in the healthcare environment, we also have like process optimization, but individual patient monitoring, like using, for example, like wearable devices for tracking, using microfluidics for that is an interesting topic, like on a very individual base. And then a larger field, it's part of life sciences, I'd say, and that's like clean meat production. So not that much like the lab automation itself, but this is a very interesting topic because it promises a large scale at the end. And now I would like to dive into like a solution which is meant more for the first two topics, like cell and gene therapies, and that is a technology which was developed by Festo for some time now, and this is our supramotion technology. And what we foresee here is like it's a levitation technology which enables us to transport goods from A to B without ever contacting them, which has, of course, massive advantages for sterile environments, for example, or if you need partial clean room requirements, for example, in an entire process. This technology enables to transfer biological samples from A to B, as I said, like in a non or in a contactless way. And this technology is pretty interesting. If you go to the next slide, we can see like in one application example how that actually works or could work together with different other automation technologies and products we have. And this is like a process which covers the entire process of like filling a biological product. In this case, we had the our process was a microbiome therapy, and there are certain containers which have to be filled with exactly that kind of therapy before being dry frozen. And these therapies have to be handled and filled in a sterile environment, in a clean room environment, because like the therapies are getting in touch with the ambient environment, with the ambient air, which makes it critical here to move things like into the sterile environment, but also get it handle it within the sterile environment without any contact. And the principle behind it is like a supraconductor, which is like frozen down to a certain temperature. It's about usually like 70 Kelvin. And the magnet on top is being frozen into that position, more or less, sticks in that position, which enables us to be rigid, depending on the geometry of the magnet, to be fixed in the position in the room, no matter like how we tilt in the end our good which we want to transport. And if you go to the next slide, we have a nice animation for that, how the process could look like. So apologies for having the first slogan in German. It's like the supramotion in a lab automation environment as a translation. And what you can see here is like the lower block is like a scale which is moved. We have a cooling element, a cryo cooler, which is gone down to those 70 Kelvin. And on top we have a magnet conveyor, which can carry like a certain load, and this can be brought into like a sterile environment. And we see here a decapping solution, and then like a dispensing solution to fill the therapy in there. We can close it again, so we are like in the sterile. We have it covered again and sealed, and now it's running to the next process, which is basically the dry freezing. And we actually built that, and as you can see here, you can levitate this carrier through all like non-magnetic materials. Of course, if you have something which is magnetizable within, then you might get a problem in sticking there, or like interfering in the height, like the air gap in there. Yeah, and this is just like one application. We have several other applications, like for example, like the weighing is one thing. We can also transport heatless here. That's very important. So we do not induct any heat by the magnetic carrier here. And yeah, this is yeah, like a pretty interesting technology, and it operates like on a very low energy consumption here. And this is one of the technologies we are working on at the moment. And Ross, if you go to the next slide. We just had some outlooks or further challenges, in particular for lab automation, which we see. And that is very much, of course, from a perspective of a component manufacturer. And one challenge, of course, is like the first one on the left side, the integration of components and subsystems into actual automation, because these topics, for us, it's like daily business, being in the automation sector. But of course, for like, that's the next challenge for biologists, for chemists, and so on. Integrating and automating stuff is sometimes like a huge hurdle. Usually you need engineers supporting them, and those engineers have to understand, on the other hand, like a lot of the biology here. And I think that lowering the hurdles of accessibility of automation is like one key point which we have to address in this sector to make it easier to automate at the end. Yeah. And then referring again to the cell therapies which are upcoming, the automation for regenerative medicines or individualized medicines, I think that is one point which which keep us busy in the automation part, for quality control, for example, but also the liquid handling containing cells, very small flow rates, and then of course like fluidic and mechatronic function integration. That will be something which we will have to deal with for the upcoming years. Yeah. All right, and at this point, I would love to close the presentation part. Thank you very much for.
Thank you so much. That was a fascinating overview. I have many questions here from the audience, so thank you everybody for your input. Keep them coming. I will try to compile them on the fly to get an accurate participation. I'm going to jump right in because we're a little bit tight on time, but also we can go a little bit over time, and please spare or hang in there with us. So I'll just start jumping in. So first question is, what were some of the challenges of the past that became easier to accomplish today through technological developments?
I can take that, unless you'd like to be honest. Basically, everything in today's world has been getting smaller, more mobile, and easier to manufacture. With developments in technology that help us manufacture new solutions, along with working with experts and scientists in the field, we're able to develop more efficient and more costly solutions moving forward to future applications. 10 years ago, if you told me that there was going to be lithium-ion battery technology, I'd question it, but that's just how technology advances, and we always look for the new cutting edge of technology to help us develop new products.
And specifically to that, what are the challenges today that you look to solve in the future?
Yeah, I'll take that. So definitely, many of the challenges are that these systems, as we've seen them, so Sid showed a lot of the liquid handling, for example. We have a lot challenges in particular the lab automation, the complexity of the processes at the end, and then integrating those into FDA-approved solutions. That's definitely one of the larger challenges, but also, for example, leverage technologies like machine learning or something to make sense of those more complex systems and use a different approach to control them at the end. These are
Small
those.
challenges up ahead.
Exactly.
Fantastic. I have a community-focused question here. So how does a community continue to aid in the advancement of technological developments, and who are the drivers? Big
That's
questions
great.
here.
I'll take that. Getting involved in different organizations like SLAS, which is Science Laboratory Automation Showcasing, or Laboratory Robotics Interest Groups. There's many communities out there. The drivers of this technology are going to be the machine builders, the contract manufacturers, the engineers, the component suppliers, working alongside the drivers, who are you, the people who attended this presentation, scientists, biologists, engineers, and interested parties in inventing.
May I add to that? So the communities interacting in real life is, of course, the first way to go. But what we also see is, like, on the technological side, there are some advancements also for online communities, technology communities, where, for example, open source for lab automation is pushed. And I have the strong feeling that this is also a way to go here, to make automation more accessible, also in very early stages of academic research and drug discovery.
Great. I think you've answered a little bit of the next question, which was where the future of the industry needs to go. I don't know if you'd like to add anything to that, but what do you see the need of the industry going in the future?
Yeah, I would like to add. So, of course, so there's two aspects to it. Like, one is as Johannes mentioned, the open source, the technological community-based aspect to it, where we can democratize it a little bit by providing some standardized software packages, drivers for the open source community. Our users can test it in the academic space, as well as the modularization, which I touched upon a lot during the liquid handling section as well, but making it as easy for our user to put together a system using something as simple as an online tool to put together a system similar to as we put like bricks together. So yeah, I think getting it down to that level is something where the future of the domain lies.
Great. So I have a technical question here. So how compatible are your technical solutions with decontamination requirements such as VHP?
So I can take that. So at the moment, most of the Festo products are like considered as clean. We have some prototypical products which are also going into the more or where we did some cleanability studies behind it. But this is usually something which we can evaluate like in customer solution projects where we can come together, and then we have some experts who can also assess the risks behind it. And that's an invitation to get actually in touch with us, because it very much depends on the application.
Great. Okay, I'm going to take a step back here from the technical questions and go, what role do you believe robotics will play in the future of healthcare, and why is now the right time for all of these solutions?
Yeah, I can take that. So we've seen, in particular in the Healthcare Catalyst program, also the research we did over the past years, is that a lot of the technologies, in particular, so if we go, for example, for healthcare robotics, a lot of advancements towards collaborativeness have been done. Yeah, a lot of questions have been solved, which were driving the acceptance, I think, of that. And on the other hand, so like this for the robotic space, for the more automation space, which might be maybe a bit simpler robots, for example, like Cartesian robots and stuff. So the automation itself, I think this one is like getting up. Of course, it had a peak during COVID, but when I see all the different like new therapies which are upcoming, they are working in a totally different way than, for example, like small molecule drugs worked in the past. Yeah, it was pretty easy to scale up pretty quick because it was like one kind of molecule to produce and then just get it out. If we're looking into individualized medicine, we will have a way more complex manufacturing process, and physical labor or manual labor in there will increase or decrease the ability to scale, of course, and also increase like the rate of errors enormously. So therefore, I think lab automation, in particular in those fields, has a very, very high, like there is a high need to automate in the end to ensure product quality and being able to scale globally even.
I'll add to it as well. We're biological beings ourselves, so the accuracy that can be obtained from robotics is a magnitude higher than what could be affected by a human. Throughput can be introduced and improved, and some of the viruses and diseases that we are researching and developing now, as you know, we're susceptible to it. So it's a lot safer, there's a lot less contamination, and a lot more accuracy when we bring robotics into the field.
I think you're addressing some of the later questions as well that were provided here. So I think I'm going to take a trend question here. We would be remiss not to mention AI. So how does AI enhance robotic systems in healthcare, and where do you see the biggest opportunities for growth in that particular section?
Yeah, if I may. So I will take the Festo perspective again, because I think there are very large opportunities, for example, in the healthcare, in the optimization of healthcare. Overall, like more in the healthcare system, so not so much in the automation. But this is something which is like actually far away from our portfolio. So what we see as an automation company is like exactly that, the control of pretty complex systems, yeah, which helps us, like where AI helps us together with closed loop technology to actually control more complex systems as we're capable, or as we were capable in the past, or maybe just due to the effort which is behind it. But what we have also seen in many applications is like that the problem-solving abilities of robots, whether it be like a Cartesian robot or six-axis robot or something, is getting way better, also with almost unknown problems, which means, or with similar problems at the end, which enables scaling again. And therefore, I think the promise here is definitely the adaptability, the robustness, in particular, when you go into organ manufacturing, you will have like your product will be different a bit each time. And maybe you still have to print stuff on top. And if you have a robotic system which can adapt to that, that will enable scale at the end.
Yeah, so just quickly adding a point there. I know we are a bit short on time, but also, so in what I also see is that apart from the, let's say, for example, working in unknown operating environments such as a bioreactor, or for working in organ manufacturing, AI could also benefit robots. They are learning different surfaces of the substrates being handled. So it could be like through haptics, adaptability and compliance-based control schemes, while also resolving kinematics in an unknown operating space. So LLMs integrated with robots is something also which could open up a whole new dimension within this problem space. So
Great. I think we have time for 2 more questions. I'm going to make the next one another technical question that just came through, and then we'll close. When designing a complete system, can you use any control software, and what are the most important features of said software?
I can take that. We try to get as close to the original application or the design as possible. We're very flexible to work with different types of communication softwares and different types of graphic user interfaces. So while developing it, we do keep it in mind. We do have our own Festo automation software suite and a backplane that communicates to all of our components. But we're also flexible and able to integrate any other type of component in a hybrid sense to be able to achieve the application's goal.
Great. And I think just because of time, we'll have to do the final question. I'm thinking big picture here. Where do you see robotics evolving in healthcare over the next 10 years?
Tricky one for sure. Yeah, I think robotics will play a key role in the scaling, as said before. So advanced skills for handling processes which are like not as standard. Like we produce valves. Yeah, they are pretty similar for each piece. Yeah, so that's a kind of automation which is classical. Yeah, you have one gripper doing that all the time, and the robustness, the use of robotics to fill the gap between manual work on the bench, yeah, pipetting and stuff, and then scaling up seamlessly over a certain range of production volumes, and then being in a high level or in a high throughput production. The space within between, this will, I think, a very valuable space for the future to enable different kinds of therapies which are upcoming. And yeah, even if they are for specific or for rare diseases or for like large, like very broadly distributed diseases, I think that could be a very interesting field to make it affordable to automate all of these solutions at the end.
I'll add to that as well. Interesting personal story. My uncle just received a knee replacement surgery that was AI assisted using robotics, a robotic surgical device. So in the future, I think it's going to enable us to have talented doctors from around the world, say, operate from India on somebody in Boston with more precision and accuracy, and assisted by AI to complete tasks, difficult tasks such as heart surgeries or brain surgeries, which will eventually help us evolve and survive longer with all the new challenges in the oncological fields and in different types of diseases and viruses that will pop up in the future.
Great. Well, with that, we're right on time. So thank you so much, Johannes. Thank you so much, Ross. Thank you so much, Sid. Thank you, all of you who've joined us today. We will, as I mentioned, have this up online on the Mass Robotics website as soon as possible.